Thank you very much for being here. I'm so sorry for the delay in your talk.
No problem.
So, Lucia has made her undergrad and master in bioinformatics in the University of Germany. She did her PhD in cooperation with Fiocruz, Brazil. I see that Uruguay and Brazil have a close relationship apart from a very large frontier, and the Pasteur Institute from Montevideo. She is currently focused in human genomics and medical genomics. She was teacher in bioinformatics and to the aim of strengthening in the field in Uruguay. And also Lucia has been teaching of algorithmic in a Master of Bioinformatics and Programming Computer. She tells us as well that she's the mother of 4 kids. You are very brave. I have to tell you. Don't know how do you do. And at last, that she likes to play handball in your free time as a hobby. I don't know how do you have free time after all of that, but congrats. So we
I
are.
like Spain.
Yeah, you have to be very well organized, I think. So she want to talk us about advances in human genomics in the Uruguayan scientific context. Diana, I leave you with
Great.
the talk.
First, thank you very much for giving me this opportunity to talk here. I'm very happy to be part of this community, really. I will talk today about human genomics and the advances we have made in our group at the Pasteur Institute in Montevideo. So these past 10 years, I have been actually working on very different things like microorganisms, yes, parasites, rodents. I have been developing tools, and only in these past 5 years, I was able to focus a little bit more in SARS-CoV-2, of course, because of this pandemic that Diana told us before, and I was working with her at this Frontera group. And also I have been working on human. This is something I wanted to do for quite a while. And I think the curse of a bioinformatician is that you jump from several different topics. But I was able to focus on human population genomics and medical genomics in the context of this project called Eurogenomes. The Eurogenomes project was coordinated by the Pasteur Institute in Montevideo and in cooperation with the Seoul National University and Macrogene also in Korea. And the main goal actually of this project was to build local capacities in human genomics. And this was done in this past year successfully. And scientifically, the project was organized in 3 phases. The first phase was a more anthropological one, where we studied individuals with specific ancestries. Then a second phase, where we studied the general population of Uruguay. And then a third phase that was more related to medical genomics, and especially rare diseases in children. So I will talk a little bit about the results of each phases, and then talk a little bit about the perspective of our group here. So regarding the first phase of the project, we studied individuals with known Charrúa ancestry. Charrúas were the indigenous groups living in our country before colonial times, and there is very few information of them. And also I have put a quote there of Galeano, an Uruguayan writer, that says, if the Mexicans descend from the Aztecas and the Peruvians from the Incas, we, the Uruguayans, descend from the boats. Okay, and this means that we think that we descend from European people. And this is like a general belief in the Uruguayan society that we think we have no indigenous ancestry. Right, and because of this, and because also of the few information that there is on the Charrúas, we wanted to do this phase, just to answer some of the question, if we do have indigenous inheritance or not, and to shed some information for Uruguayan history. So what we did was HiCov, a whole genome of individuals with known Charrúa ancestry. And then what we have done is local ancestry estimations, that is, you can infer for each part of your chromosomes to which ancestry it most likely belongs to. So here you can see, you paint the chromosomes according to the ancestry. Here in red, you can see the European ancestry, in green, the indigenous ancestry, and here in blue, you can see the African ancestry. Okay, and for example, in this individual, you see a lot of green, so you see a lot of indigenous ancestry in their genes. And this repeats for the other individuals as well. And you can, of course, quantify the green of each individual. I will show this in a minute. But also what you can do is take these tracts, these ancestry-specific tracts, especially the green ones, now we are interested in the green ones, and compare those portions of the genome with other tribes in the region whose genomes are available, right? So we did a PCA here with our Charrúa individuals, only with the green portions, and compare it with the other South American tribes that were available at that time. And we see here, we have here the descendants of Charrúas in the asterisk, and we see a genomic affinity or a similarity with these groups that are Amazonian groups. Here we see Guaranis and Caingangs, and here we see a similarity with the Diaguitas, which are a tribe that were here in Argentina and also northern Chile. Okay, this similarity here with the Guaranis were actually kind of expected, since there are historical records that reports Charrúas and Guaranis coexisting in what they're called missions, Jesuitic missions, for example, that are indigenous settlements that were organized by priests, European priests, that came here to impart like Christianity and so on. So there were reports on that. But the Diaguitas were something more unexpected, because actually geographically they were not as close to our Charrúas, but nevertheless, asking a little bit more and talking with archaeologists and anthropologists, we found out that the Diaguitas suffer from a great geographical expansion, so they were able to go towards Uruguay as well. And also the Charrúas, they were reported in some records that they were also present in somehow this region in Argentina. So actually this is also plausible, right? And additionally, something that I found interested in is that this population that we studied is very aware of their indigenous ancestry. So they know that, and together with the anthropologists that worked in the group, we are able to build the family history of each of them, and we can estimate a self-declared Charrúa proportion for each of those individuals. And we can also then, this is here in red, okay, the proportion of indigenous ancestry. And then we here have the actual genomic proportion of indigenous ancestry in those individuals. That means the quantity of green in their genome. And we can see for each individual, or for almost all, there is much more genomic indigenous ancestry measured by the genome as what they knew, even in this population that they really know and they are really aware of their indigenous ancestry. So I found that interesting. Then we tried to see if we found selection traits in the genome. And we did IHS only on the green portions of the genome, and found some regions here that were significant. Yes, for example, I just pointed out here the chromosome 1 region, this one, and the chromosome 4 region, this one here. So we see in the chromosome 1 region, we see here the Charrúas in the bottom, and we see a long blue haplotype present in almost all of the Charrúas. And also when we compare it to other natives that we had the genomes also, we see this haplotype also conserved or present in those other natives. So maybe this is a signature, a signal that is older or common to other natives as well. So we see here the genes that are present in those regions, for example, the actin-2 gene and the MTR gene, that is a methionine synthase gene. And in the region 4, in the chromosome 4, for example, we see also Charrúas with this long blue haplotype, not all of them. And here in the other native, we don't see the whole, or almost not, we don't see the whole haplotype. So it is a portion of the haplotype there, and then it was broken up due to recombination probably. So maybe this signature is more proper to the Charrúas. And what we see here is this gene that is related to the innate immune system, and also this other gene here, SPCS3 gene, that is related to the virion production of flaviviruses, right? Flaviviruses are such as Zika or dengue, or some viruses that could have been present in pre-colonial times in the region as well. Great. So, and then something we wanted to address is if we can infer from the genome when was the contact between the Europeans and the indigenous groups, and when was the contact between the Africans and indigenous groups. So not the contact that they saw each other, but the contact with admixture. So we wanted to date the admixture. And this can be done also from the genome and taking these ancestry-specific tracts that I told you before. If we take the length of the specific ancestry tracts, we can fit exponential distributions on those fragments, and then we can infer when these both ancestries coexisted and left descents, right? And we took this information and tried to fit those models, and what we found was we found a first wave, a first contact from Europe to the indigenous groups here in our location that was dated in 1660, okay? And this concurs with the Franciscan mission, the foundation of this Franciscan mission, what I told you before about those settlements. So this could happen that the Europeans came, they created this settlement, and then it could happen that the admixture happened. Then we found out a second wave or migration wave or admixture date of the Europeans and also Africans with the indigenous people, one generation later, 1680 more or less. Of course, these dates have uncertainty on it, right? And 1680, this concurs actually with the foundation of Colonia. This is a city here in Uruguay. And according to historical records, also this city needed a lot of soldiers to work on Colonia, on the foundation of Colonia, and also a lot of men work. So yes, a lot of slaves to work in the city. So it concurs with that. And additionally, there is another wave of Africans that had mixed with the native in the 1700s, and this concurs with the foundation of Montevideo, roughly. And this is a port city, the capital of Uruguay, as Diana mentioned before. And this city required a lot of work also because it was a city port with a lot of dynamics, so many workers were brought to work there. So maybe this concurs with that. So I think we were able to put some facts or shed a little light in the Uruguayan history regarding the charruas. And also we were able to see that there is still indigenous ancestry in the genes at least from those individuals that we studied at this phase. But actually, can we see the same in the general population? Because this other population was targeted, was looked for, and we knew they had indigenous ancestry. But in the general population, so we sampled 30 individuals representative of Uruguay, and we did a high-COV whole genome sequencing again, and we found that there is a 14% of indigenous ancestry also in the general population, 78 European ancestry and 8% African. And additionally, what we found is that when you do a PCA of the 30 individuals we have here, and we see the red dots are individuals with less indigenous ancestry, and blue is high indigenous ancestry. We see the PC1 correlates with this proportion of Amerindian ancestry, separates the individuals with low and a lot of Amerindian ancestry. And also we see that there is a correlation between the distance to Montevideo and where the people were sampled, right, and the Amerindian proportion. So the further away you are from Montevideo, the higher the Amerindian ancestry is expected to be, right? And this is only preliminary results from the phase 2. We're working on expanding a little bit more the sample and so on. But this is actually already a little bit interesting. So now going from the general population or population genomics to specific patients. And here, this is the phase 3 of the project. And I want I picked 2 cases of medical genomics that I want to share with you, just to, share the importance of medical genomics. But there are many more cases that we could talk about. So this is the first case we did. That's why I put it here. It is actually not whole genome sequencing. This was a mitochondrial DNA sequencing. And this was a girl with 2 years old. She presented mobility problems. Half a year later, she was not able to walk alone anymore, and she presented severe and progressive neurological anomalies. And the question was if this disorder was Lake disease. So what we did was we sequenced the girl, the unaffected sister, and the mother, the mitochondrial DNA of those. And we found several variants. We were able to filter some of them. And then we kept these 3 variants that we believed they could be probably damaging. And actually we kept this variant only as that we believe it is the causative mutation. That is a variant in the NDN1 gene, which is the complex 1, which is the largest of the 5 respiratory complexes. It has a high heteroplasmy. Here we can see it in the patient, and low in the mother. This is the mother here, and low or intermediate in the sister. So that's why we kept this mutation, actually not the other ones that had the same heteroplasmy in everyone. And then also this position in the genome is highly conserved according to evolutionary studies. So changes there could be impactful or could be pathogenic. And also we have here, we see the protein, here the change, amino acid change. The change was a serine, which is a polar residue, and it lies right in the entrance channel of the kinome. Okay, and this could impact actually the entrance of the kinome. It is just a hypothesis. So maybe, but just to mention that it could be important. And then also we culture the fibroblasts of the patient, and we see here, this is the complex 1. This is the concentration of the patient of the complex 1, which is lower now here in the patient. But after we added coenzyme Q10, we were able to restore the concentration of complex 1. So actually what we found, we think we found is a mutation that explains the Lake disease of the patient. And also we proposed back then specific concentrations of coenzyme Q10 to improve the symptoms of the patient. And it was okay for a while. Okay, this was the first case, and then a second case, and this is whole genome sequencing. This was a girl with 13 years old. She presented at the age of 6 hypertension and dyslipidemia, and she had a renal artery stenosis, with right renal artery stenosis, and she was diagnosed with renovascular hypertension because of that. But then other stenoses appeared in carotid and vertebral, so she was characterized with a panvascular alteration, so more general. And she had previous molecular studies made already, cardiovascular polymorphisms because of the hypertension and also mitochondrial DNA, but no alterations were found. So what we did is whole genome sequencing of the girl. And what we found was this mutation here, which is a frameshift mutation that had all the flags of being pathogenic. It causes a stop codon almost immediately. It has like 0 frequency in the general population. It was predicted as pathogenic. It lies in this gene, YY1AP1 gene. And this gene was related to the Grange syndrome, which is a very rare syndrome. And it is a recessive syndrome. So actually this only variant couldn't explain completely the phenotype. So what we took a look after that is if we found some structural variants that could also impact this gene. And actually you can see it without any structural variant algorithm that we did, of course, but then you can see it almost by eye here. If you zoom rightly the IGV view, you see here that there is a deletion of these 2 first exons of the gene also in the girl. Okay, so 2 exons or 1 exon depending on the transcript. So then we analyzed the parents, and we saw that the father has the frameshift deletion and the mother had the big deletion. All of them are unaffected, but the girl is a compound heterozygous for this gene, and she presented this Grange syndrome. Also, the Grange syndrome has many symptoms, and some of the symptoms were not looked for in the girl because the Grange syndrome was not as an option. But now that it was, they found additional symptoms that fit very good to the Grange syndrome. So we were pretty confident that we found it here, the causality of this phenotype. Great. So Actually, in these past years, we have sequenced several cases of whole genome sequencing. We have also a lot of mitochondrial DNA cases analyzed. And I have also a startup company that does whole exome sequencing for diagnosis of rare disease. And we have here also a couple of cases. And almost every time the diagnosis success is like a 50%. That is, in 50% of the rest of the individuals, I'm not able to do a diagnosis. And this is okay. This is what the literature expects, and this is what is happening nowadays. But we wanted to see if we can help improve this diagnosis. So we are working actually with several students at different levels of this problem, just to see if we can contribute a little bit to improving these parts. So we'll show you just a couple of work that is ongoing with our students. This is a master student of ours. We built a prioritization classifier for non-coding variants. You know, when you're working in medical genomics, or in general, actually non-coding variants are always a headache because they could be important, but they are very difficult to assess their importance. So it's not the same as the coding variants, right? So we are building a classifier for that. And here, okay, here we downloaded CleanBar data. So about 100K variants with a label of pathogenicity. So we have 5 labels that we use: pathogenic, probably pathogenic, variant of uncertain significance, probably benign, and benign. Those are the 5 categories. So we download variants with those categories. Then we annotated those variants using ANNOVAR. So we kept only half of them, more or less, of the variants, because also label imbalance or quality issues and so on. And then we imputed missing features of ANNOVAR. We tested also several different algorithms to do imputation there, and we used KNN afterwards for that. And then we did feature selection on this final data set. We kept only 13 features of ANNOVAR, and then we did gradient boosted trees to do the classifier. We are actually here at this part when we are looking at a validation set, an external validation set, that is a set of non-coding variants with labels on pathogenicity to present to our classifier and see how it works. And after that, we will be able to present this classifier to new patients to assess and prioritize non-coding variants on these new patients that we have. Those would be patients that the exome or the coding variants wouldn't give us the answer, right? This would be for the difficult patients that we were not able to classify or to diagnose yet. And of course, the results will be seen with a geneticist without a doubt. So this was one. This other thing is I have built a couple of years ago a platform for the interpretation of variants together with our group. And what you can see in this platform is pretty straightforward in this sense. You have the variant that you find here. You have all the information here that you need to do a proper classification, like all the OMIM associations, CleanBar data, the frequencies in other populations, in silico predictors, quality stuff, and so on. And also you have a ACMG classification. You know, ACMG is the American College of Medical Genetics that had set a lot of rules to classify your variants in these 5 categories as well. So we have this classifier there inbuilt, and sometimes, or more often than not, you have variants that are still of uncertain significance after you have done all the things that you can do, okay, after seeing all the information and so on. For those variants that you still don't know what they are, we have built some extra features here that is based on natural language processing. We have download several abstracts of PubMed. We built a corpus there, and then we marked entities of genes and variants and diseases using ontologies just to rapidly scan each abstract and see where your variant or a gene or the gene comes together in the same sentence or at a distance with a disease, so that the geneticist has all the information that he had before, and then all the abstracts that are relevant to the variant that he's looking at that time. So it is faster for him, and it looks for relevant articles in a glimpse of an eye. So this information could help then the geneticist to do a proper classification. And the last thing we have been doing, it's not quite done yet, is the work of a student of ours, a master's student in bioinformatics. She's building a platform for learning and classification of variants. So the thing here is, let's say we are here and we are trainees that we want to be trained to classify variants. So we have a low score because we are not that good in classifying variants. Our hope is to be upgraded to here, to the multiple expert panel, and to be very high scoring classifiers of variants. So if we are good classifiers, we get a good score in the platform. If we are good, we are presented, this is my platform here, we are presented with variants that are complicated, that are contradictory, that the labels don't fit, or something is complex in this variant. So we are presented to these variants. And the multiple expert panel tries to classify that using a lot of things, using all the annotations of OMIM, other database of ANOVAR, other databases that are available, and also an ACMG calculator of classification. After that, if they can classify that, the variant classification is stored, meaning we have one of these 4 labels here, and it is stored with also the score of the expert that classifies that. So this information of the variant with the tag of benign, likely benign, likely pathogenic, or pathogenic, together with the score of the expert, is fed again to the platform. So the platform will get high quality data to build the classifier. Now the classifier is built with the tree, with a gradient boosted tree as well. So we have a good data set to build the classifier. And then new data can come. So a new variant can be presented to the database. And if it is a complex variant, or if the platform cannot classify it by itself, it is presented as a variant, a complex variant, to the panel of experts, and we try this all over again. So this is quite a nice platform that Camila is building. So in a few months, it should be ready to go. So, and what is now the plan for our group in these following years? We still want to do population genomics and medical genomics, of course, and then we wanted to, or we are adding this third base here, which is ancient DNA. Here we are already working with, we have an ongoing project, new ongoing project with the Faculty of Humanities, with the archaeologist Lopez Mas, with the University of Tübingen in Germany, and the Max Planck Institute also in Germany. So the archaeologists here have identified 3 archaeological sites with 3 different groups, indigenous groups that they were there. And we are trying to extract DNA from the remains and try to describe those indigenous groups. So we already are in this part. We have identified or extracted DNA of these remains, and we are waiting actually for the results of the sequencing. So we are very happy to find out what the results of that would be. So, and then regarding population genomics, we have a lot to do on the phase 2 of the project that I told you before, but also I wanted to mention this, that we are part of Latin Genomes. This is a group that is supported by the Broad Institute and the ICDA, which is the International Common Disease Alliance, but is led by Latin American researchers, and the main goal here is to increase the representation of admixed individuals in GWAS studies. You know, because in GWAS studies, which are very expensive to make and so on, there are only very few Latin American representation there, or admixed individuals in general. So, and this generates a gap in the application of genomic medicine afterwards. So we want to narrow a little bit this gap. So we are part of this project, or of this Latin Genomes group that tries to focus and improve the representation of Latin American individuals there. And finally, genomic medicine is also what we want to pursue. And we have like several different genetic diseases that affect the Uruguayan population that we are working on. And those three are mentioned here: Behçet disease, segmentarian focal adenosis, and complex congenital cardiopathies. We have also students working on that. So, and of course, I am a bioinformatician, even though it sounds like I'm doing only genomics, but I really like also developing tools and stuff. So we are still trying to develop some tools, one regarding mitochondrial DNA analysis, and the other one I just mentioned about the non-coding variant. So we are still trying to improve or developing tools to get out there. So, right, so actually to finalize only, I think it's important to include this part in the work to understand a little bit the past that we have. And also this past could share some light into the present population that we are dealing today. And also this could give us like more evolutionary perspective on the diseases that we can face in the future. So I think these three topics will improve our work. So thank you very much.
I cannot open my camera now. Thank you very much, Lucia. Such a nice work. It was a very nice work to see this you've done with the ancestry of our native people to recover our history. As you say, we are not descendant of the boat. Of course, we are not. So it was very nice to see that you are trying to recover our history, genomic history. And also I was thinking about if that can be applied to diseases which are local diseases and they might be different from Europeans one, and if this eventually can suggest some treatments. This is.
I think that the programs first step towards understanding afterwards the genetic diseases. There are some diseases or some variants that causes diseases that are different, the ones that are happening in Uruguay, the ones that are happening in Europe, right? So that is the problem, what I mentioned before on these Shiwa studies. And I think I see a question there also, that when you do these big studies and you describe, I don't know, European population, and then with a disease or not, and then you compare and do this association analysis, you found then regions or you find variants associated to that disease. And then when you want to diagnose new individuals from other populations, you don't see this variant probably related to the disease. Maybe it is the same disease, but this variant does not explain the phenotype of the patient. So then if you don't do the studies with the right population when you want to apply afterwards the medical genomics diagnosis, it normally doesn't work that good. And maybe we thought before, Uruguay, it's okay because it's so European and it's okay. Well, it's not as European as we thought before. So, which is great, but also we should do and we should have a representation of admixed individuals in the GWAS. This happens for Argentina, for Brazil, for Latin America in general. So I think this group is really cool in this sense. And it's Latin American researchers doing research for Latin America. So I think it's pretty awesome.
This is nice, yes. Nice to see. I have some questions here. This is also a gender perspective in genomics medicine is important, isn't it?
Sorry, can you, the gender perspective, yes, also. In genomics and in general, so there are like several phases there, right? Because there are some diseases that are women that are less studied. This is already one inequality there, right? Because of, I don't know, funding or interest or whatever. And then it's true that sometimes it's not even considered gender. Or the experiments or sometimes animal models or whatever are not taking into account this part, and it should. So I think it is also important. Yes.
You can put in the near future arrow.
Yes.
So there is another question here, but I think you already answered some of it. It says, have you, I have a question for you. Is there any possibility of checking the prevalence of some genetic diseases associated with those genomes and the difference between those diseases coming from Europe?
Yes.
I think it
Yes.
was.
So the study here, or what was good in this whole Eurogenomes project, it was okay. We built human resources and researchers and so on, but also we made a small, modest database. Of course, we cannot compare our numbers with the Argentinian or Brazilian databases, of course, but we built like a reference panel of the Uruguayan population. And maybe it has only 80 individuals, but nevertheless, you can check for since it is whole genome also, you can check for the GWAS variants, for example, that are relevant for the European population, and you can check them, the frequency of those variants in our database, and you can see, and it's something that we wanted to do, you can see the differences in the frequency of those variants. Sometimes variants that are pretty rare, theirs, hence probably maybe causative or something, or could be considered like, yes, rare, they are frequent here, or the other way around. So yes, what we built is actually this small reference database that everyone can consult, right? It is open. So we can share this information, at least the summary statistics on that. So we can compare with other frequencies in the world. Yes.
That's nice. I would like to make you another question. Everybody's telling you such a nice talk, inspiring, enlightening. So you have many congrats from the people. And this person is asking, which is the difference between your platform and the availables in internet as Barsom or Franklin?
Yes, great. So the platform we have developed has a lot of information that is already there in Barsom, Franklin, Bloomberg in general, or others that appeared after that, or me, and so on. What it had is you can check the frequency of your variant, not only with the other populations in the world, but also with the local Uruguayan population. And also you have a way of post-processing these articles and so on, that this natural language processing part, that this is not part of the other, that I know of the other platforms. And also you can do automatically a report if you want. So you upload the annotated BCF file, then you do your filtration and your classification and do the NLP part, and then you can directly inform to a PDF with your findings.
Yeah, it adds some possibilities. So I think we'll do the last question because we are very late already. Thank you very much for your talk. Say, do you think that all the mix of ancient lineages create new variants and mutation of some diseases?
I think so. The admixture of populations is always a great thing. So you mix a lot of different variants and different frequencies. I think always very good for genetics and for the population in general. And if it's not about the mixing of ancient lineages, but there are some characteristics in ancient populations, for example, or in the indigenous population, are characteristics, maybe not diseases, just mutations or characteristics that were able to pass along to the present, to the modern individuals. And this is this we can see the parts of the genomes that are actually from indigenous descent, and which are more recent and which are more ancient. We can see it. We have like a 2%, I don't know, Neanderthal ancestry in the European. So you can see that. I would not associate this to diseases or something. It's like the variation of the mutations without a negative connotation, right? As variants. The variants from ancient or indigenous alleles can be present in our own genome, yes, but they're not linked to anything in particular.
So we would like to thank you.